Hello everyone, this is Dr.SHIVA Ayadure. This is another one of our videos, podcasts for this morning. We had wanted to do it earlier at around 8:30, but I wanted to get some more slides together. As you know, the purpose of these podcasts is to be educational and to give you a deeper perspective on extremely interesting topics, sometimes controversial topics. Today's topic is genetically engineered foods, or GMOs. And the question is, first of all, what are they and are they safe? Most of what I'm going to be sharing with you here comes from my own research. It's original research that we've done and we've also published. So I want to take a little bit of time, first of all, to give you sort of the larger perspective on what's actually going on with genetically engineered foods. And probably the first thing is to begin with, what are they? People have heard the term GMOs, or genetically engineered foods, and in fact they're used interchangeably. And most people don't know what they are. They do know something doesn't sound right about them because it sounds like they're sort of done in a lab, which they are, and it doesn't feel right to a lot of people, and other people say they're absolutely safe. People are being paranoid. But let me just share with you a slide here. What is a GMO? You know, a GMO, first of all, is a genetically modified organism. And the term genetic engineering or genetically engineered foods was also used to refer to GMOs. In Europe, they started with genetically engineered foods, which they said genetically modified organisms. And that term genetically modified organisms is used in the United States. However, some people started using the term GMOs to refer to any type of genetic modification. And they created a confusion between, for example, natural plant breeding. For example, if you think about it, for centuries or millennia, when we did farming, we bred animals, or we bred different types of corn or different crops. Well, that was what we call sexual breeding, which means you actually bred the two plants, bred the two animals. Sexual reproduction needed to take place. And that resulted in quote unquote genetic modification. So for a long time, people actually, including major newspapers like the New York Times, etc., would tell people, oh, don't worry about these genetically modified foods, referring to genetic engineering, which takes place in a lab. They're the same as genetic modification that takes place from natural plant breeding that's been going on for centuries. And this is where the confusion came in, and that's what I want to clear up. But going back to the slide, you know, in a genetically modified organism, you're actually taking genes from the DNA of one organism and putting it into the DNA of another organism. So for example, you could take the DNA of a flounder, which is a fish, which maybe there's a certain characteristic in that DNA which gives it a very protective skin to protect it from the cold, and stick it into the DNA of a tomato to make it more resistant to weather. Now that would never ever occur in nature, meaning never would you see a flounder having sex with a tomato. At least I haven't seen one, maybe you have. Obviously you may breed different kinds of tomatoes sexually, but this is asexual, which means without sex, genetic changes that are taking place in a lab environment. This cannot occur in nature. And I think that's probably the most important most important point we need to make, is that what we're talking about here is genetic changes that are taking place to an organism that occur in the laboratory environment. It doesn't occur in nature. So next time you hear someone saying, oh, don't worry, genetic modification is the same as natural plant breeding, we're just accelerating it, it's not true, because this is occurring in a laboratory environment. Very difficult to have a flounder and a tomato have sex in the natural world. The second thing I want to walk you through is how do genetic modification take place. And what you're seeing here is you have a bacteria, and you're taking the gene from this bacteria. So there's a DNA extraction isolation process that takes place. So you have a bacteria which has its own string of genes. By the way, going back to basic biology, genes are pieces of DNA which code for different types of characteristics of an organism. So, for example, a worm has around 20,000 genes, and we'll shortly tell you that's the number of genes we have, number of genes. And different sections of the DNA, so consider it as this big long ladder, and different pieces of that DNA have code for different characteristics. So for you, a section of DNA may code for the color of your eyes, another section may code for the color of your skin, etc. But in genetic engineering, we're literally taking the DNA characteristics from one organism, bacteria, isolating it, and then we clone it, which means make copies of it, and then that is then cultured and then stuck into another organism. So those of you, if you're watching the video, and you don't need to because I try to explain it even if you're listening to a podcast, what we see here is DNA is extracted from a bacteria, it's cloned, and then it's cultured, which means it's grown, and then pieces of that DNA are put into a plant. And so that's where we're transforming or taking a piece of DNA and transferring it to another organism. Here are various examples of different kinds of genetically modified foods. Corn is a genetically modified food, which means in that case a gene from a bacterium was taken and put into the corn. Soy, cotton, papaya, rice, rapeseed, or what's known as canola oil, potatoes, tomatoes, dairy products, peas, and there are others, but these are the top 10 commonly genetically modified foods. So I'll repeat again: corn, soy. By the way, 97% of the soy in the United States is genetically modified. Cotton, papaya, rice, canola, also known as rapeseed oil, potatoes, tomatoes, various dairy products, and peas. Why did companies, which are the big agrobiotech companies like Syngenta, Monsanto, which is now part of Bayer, do genetic modification? To give you a high-level background on this, a company like Monsanto, for example, by the way, Monsanto came out of the military-industrial complex, which means out of war. They, along with Dow, in the 60s created I'll just come back to video here. They, along with Dow, created a herbicide called Agent Orange, which was used to drop over Vietnam to essentially kill the foliage. So the Air Force of the United States could better see their targets below. So it literally kills foliage. It's an herbicide. They're herbicides, and that's what Agent Orange was. And they used to deliver it by air and drop a lot of it on the forest. Well, when the Vietnam War ended, by the way, a lot of that was also tested, believe it or not, on the island of Hawaii. It was used as an open airfield. And there's a movie that I did that was produced by Pierce Brosnan, directed by his wife Keely Brosnan, called Poisoning Paradise, which I'm the main scientist describing a lot of what I'm going to describe to you here. Poisoning Paradise is a movie if you want to get a hold of it. But after Vietnam, they retooled themselves, which is essentially what that means is the powers at Monsanto said, well, we've created a way to drop herbicide and kill foliage. Why don't we reuse this in the farming field? Because remember, it's another, they did something in the wartime world. Can they make money in the civilian world? So that resulted in them creating another product called glyphosate, which has been in the news. There's been a bunch of lawsuits on it, or what's commonly known as Roundup. Some of you may use this in your yard, but they dropped glyphosate from the air for factory farming. As small farms were being overtaken by large farms, the goal was, hey, let's farm thousands of acres, and let's say you're growing corn or soy, weeds are going to come up, and we will drop Roundup or glyphosate from the air. And that allowed Monsanto over a period of time to essentially get a monopoly on essentially weed control on farms. But interesting enough, the weeds that were dying is what Monsanto's glyphosate or Roundup was doing. By the way, the way you spell glyphosate is Glyphosate, glyph, like glyph, glyphosate, Glyphosate. That's glyphosate. So as glyphosate was growing, Monsanto essentially built a monopoly on this pesticide. They were also noticing that there was a yield lowering of the corn or the soy. So on the one hand, now they had a monopoly on every major factory farm using glyphosate, but they noticed yields were going down. So what they did was they said, could we create, let's say, a version of soy or corn that could withstand their own herbicide and not get killed by the herbicide, increase the yield. So what they did was they took the gene from a bacterium, in the case of soy, and they stuck it in to the soy seed, and that created Roundup Ready Soy, RRS, Roundup Ready Soy. And that process, and they did a similar thing with corn, etc. But what it allowed them to do was own the seed, because now it's a genetically modified organism. They could own that seed, and they could essentially license the ownership of that seed to the farmer. So if you were a farmer, you were growing soy, you were using Roundup, you wanted to increase yield, you also bought Monsanto's Roundup Ready soy seeds, by the way, which had a license for a year, which means you couldn't save your seeds. You had to buy the seeds every year. Very profitable for them, right? On the one hand, you own the pesticide and you also own the seed side. So this is sort of the background of how all of this came to be. One of the ways that I got involved in it, my PhD is in biological engineering. Some of you may know that. I have 4 degrees from MIT, just to give you my credentials. I came to MIT in 1981. My undergraduate degree is in electrical engineering and computer science. I've always been deeply interested in medicine and biology. In fact, as a 14-year-old kid, while I was inventing the first email system in the world at Rutgers Medical School, I was also doing medical research, applying computing to biology and medicine. So I always wanted to combine both of these fields. When I came to MIT, I was deeply interested in medicine, but found that the way that we looked at the body was almost as parts. We didn't consider it as a whole human being. So I went in and out of MIT doing various degrees in engineering, because at least in engineering you have to look at the whole thing. So I have a degree in electrical engineering. I went out and started a company, came back to MIT, did another degree in the field of scientific visualization out of the MIT Media Lab in media arts and sciences. Some of you may or may not know, I also I'm a big lover of art, do a lot of art myself. So I have a master's in that field. But concomitantly, I also did another master's in the field of mechanical engineering in what's called applied mechanics in the area of wave propagation, to understand how waves move through materials, be it sound waves, all different kinds of very interesting waves that move through material. And that was the basis of my master's thesis. Went out, started some more companies, came back to MIT in 2003. And the reason I came back, it's best expressed in the slide that relates to what we're going to talk about with GMOs, is in 2003, some of you may know, the field of biology was undergoing a revolution. Why? As you can see from this slide, what was occurring was, in 1993, the Human Genome Project had started, and the purpose of the Human Genome Project was, could we actually map out every gene in the human DNA? When this project started, we didn't really have an understanding of how many genes were in the human DNA. We thought that we had a lot of genes, and the reason we thought we had a lot of genes, and I'll explain what I mean by lot, is we knew a worm, a nematode, had around 20,000 genes. So you can see from the slide, when the Genome Project started, after various approximations, people thought we had about 100,000 genes, at least 5 times more than a lowly, quote-unquote, a lowly worm. And as you can see, as the Human Genome Project proceeded, by 2003, we weren't finding all of these genes. We were essentially lowering our estimates every year, and it turns out we only have about 19 to 20,000 genes, as you can see from the slide. So this flipped biology on its head, because in fact plants, corn, actually have more genes, believe it or not. That's what's fascinating. Plants actually have probably 50, 60, sometimes double the number of genes that we do. So what this led into was people recognize that we are not our genes. Because if we only have the same number of genes as a worm, why are we more complicated? Again, basic biology, genes give rise to proteins. Those proteins actually interact back with the genes to create what are called molecular pathways, and you get a lot more complexity than thought of. So complexity is not a function of the number of genes. Complexity is a function of the molecular pathways. So what happened was biology went into a very, very different space, and that space was recognizing that maybe what we needed to do was we need to look at the body more holistically. And at this time, Peter Hunter, who was working between Auckland and Oxford, and he was working what's called the Human Physiome Project, which is could you mathematically understand the whole human body, which meant it's not just the genes as you see on the left side here. It's not just genes that give rise to the human body, but genes give rise to proteins. Proteins interact with each other, create different cellular function and various functions within the cell, and different cells compose tissues then compose organs. Just to give you an idea, we have about 12 major organs. We have 4 different tissue types. For example, in the heart, there's around 300 different kinds of cells, and we probably have 100,000 plus proteins or more. Right now, I think we've mapped out probably around 50 to 100,000 of those proteins, but they're probably about 500,000 proteins. So the idea was this is a much more complex problem. And at that time, in the world at that time, the National Science Foundation, so in 2003, put forward a grand challenge, which was, could you mathematically model the whole cell? So if you look at the cell as a very complex reactor of different chemical reactions, could you mathematically model this? And my advisor at MIT, Professor Forbes Dewey, invited me to come back, and he said, Shiva, you should finish up your PhD. There's this field where computing and biology can come together. You love biology and medicine, and you have an expertise in computing. So that's when I decided to come back to MIT. And the way I looked at this problem was, instead of looking at it in this very large way, because you can think about the millions of types of reactions or combinations that are going on in the body, it would be very hard to mathematically model this. In fact, people were getting away from this field, even in 2003, because they weren't able to model more than maybe 50 reactions. And my approach was, why not break down the larger thing into smaller, manageable reactions, which we call biological pathways, and we knew some of them were becoming mathematical models, and why not treat the models as almost individual little subsystems, each of which could be owned and maintained by different people. And I would write a technology platform that I ended up calling CytoSolve, CYTOSOLVE, which would allow a decentralized way of integrating large-scale molecular pathway models. And that was the development of CytoSolve, which was between 2003, my PhD work. And by the way, this was the basis of my PhD work, which I published in 2007. I went on to publish a major paper sharing my findings in the development of this new invention. Some of you may know email, what I invented in 1978, was really a large-scale system also to integrate the various parts of the office communication system. Similarly, CytoSolve was a way to integrate the many, much more complicated parts of the intercellular communication system. So it allowed this very powerful way to integrate. This was published in various journals, from IEEE to Springer. And we published these between 2007 to 12 to validate in the scientific world that what we had done was noteworthy and important. We put it out for peer review. In fact, a major article came out in IEEE called Creating Accurate Models of Life, in which CytoSolve was featured. I also then started applying this to real problems. In 2012, we spun out CytoSolve as a new company. And at that time, an article came out in Nature, one of the most esteemed magazines, saying if you're going to really solve cancer, you have to use combination therapy, which means combine different drugs. An individual drug has various side effects. Maybe we could lower the toxicity. And my thesis work was featured in that very important scientific paper. I didn't know any of the authors, so it wasn't some inside job. So we raised money, spun out a company. We actually used CytoSolve to discover a multi-combination therapy for pancreatic cancer. I went on to publish various papers with my colleagues at MIT in the fields of nitric oxide and cardiovascular research, work with the US Pharmacopoeia to support the research in multi-ingredient therapy of supplements. And we, in fact, as I mentioned, got an allowance by the FDA to take a combination drug that we had discovered for cancer out to market, or out to clinical testing. Also, we did some very recent research with the University of Southern California, the Zilka Institute of Neurological Research, showing how you could use CytoSolve to integrate the various mechanisms of neurovascular disease and show commonality. So the reason I'm sharing that with you was there's a very powerful tool that we developed which allowed people to really start using CytoSolve to understand at the scientific level the mechanisms, how things were taking place. And CytoSolve is a company now which is used for research, which is used for substantiating nutraceutical products, which is actually used for discovering new combinations. But getting back to genetic engineering, what happened was the way I got involved in genetic engineering wasn't, or foods, is that someone just sent me a message here, which I'll respond to. One of the important things that came here was I was walking down the halls of MIT, and there's this magazine called the MIT Technology Review, which is one of the prestigious magazines. In fact, many years ago, the work that I'd done for automatically analyzing email was featured on the front page. So it's a pretty big thing to get featured in there. But this article I saw in there, which said, front page article says, "Buy fresh, buy GMOs." And this was essentially, in my view, it caught me because it was making fun of the buy fresh, buy local movement. And I've been a big proponent of, eating organic local foods, I think, for various reasons. So when I saw this, I was a little concerned what this was about. It almost looked like an ad for something. And as I read it, the article was really about how the entire world needs genetically modified foods or we're all screwed. That's where this really starts at, that all the poor, brown and black people in Africa and India need genetically modified foods. And as I went through this, some of it didn't make sense. And so what I wanted to do was really understand what was going on here. And I recognized that there was a debate going on in the world. There were people who were pro-GMOs, a lot of the big agrobiotechs, some scientists, your typical academics. And then there was a non-GMO movement, people, essentially civilians, saying this doesn't make sense, they didn't like it. And I really wanted to go beyond this left and right or pro or non. And that's really part of why many of you know I ran as an independent, because I felt both parties essentially take every issue and they split it into pro or against, and they never really want to go for the truth. And as a scientist, what I'm interested in finding out what the truth is, and the truth is actually much harder to find out than having an opinion. A lot of people try to have opinions, like you see here, and the idea was, what is actually going on? Could we really understand this? And as I started doing the research, the issue is there a middle ground to this? And I noticed that RT, which is Russian TV, there's sometimes the US news at this point was basically telling everyone that GMOs are absolutely fine, not to worry, don't worry. Genetic engineering is the same as natural plant breeding. In fact, writers for the New York Times are saying this. And that didn't seem right, because it seemed, as a biologist, you're doing essentially asexual reproduction of genes, very different than actually animals having sex or plants having sexual reproduction. And this article caught me around this time, as I was looking, it said, study reveals GMO corn to be highly toxic. And the article actually talked about, and this was an RT, which I have a lot of respect for because they cover stuff that we in the United States don't cover. And the article basically went on to say that people were believing, and I'll read you the claims that there's no difference between GMO, non-GMO corn are false, says Honeycutt, who adds she was floored after reading the study. Essentially, the notion was that, don't worry, a genetically modified food is no different than a non-genetically modified food, that there's absolutely no difference, that GE foods are the same as non-GE foods. And so that made me wonder, how was a genetically engineered food being allowed into the market? Like, what actually goes on here? And as I probe that, the question is, what is the difference between, let's say, a genetically engineered tomato and an organic tomato? So you have the non-GMO tomato and the GMO tomato, and the question was, what is the difference? And it almost felt like, and I'm sort of going back in history here, that people had put forward the idea, don't worry, they're the same. And that's what was being brought out by this GMO corn people had found, which had high levels of formaldehyde. Farmers had found a bunch of corn where they measured it was much higher levels of formaldehyde than the non-GMO corn, and no one really knew why. So you could say that, and the non-GMO or the pro-GMO people are saying these people are just alarmists. There's nothing really here. They're crackpots, and the people who are being called crackpots were saying we actually have found some. So anyway, there is this debate, and so as you look into this, you find out that the real issue is, what is the difference between a non-GMO and a GMO corn? It's like asking, what's the difference between David Banner, the Hulk, and the Hulk, when he transforms. And as I went into this, I found out that around 2000, the Food and Drug Administration, which was the foods commissioner at that time, was a guy called Michael Taylor, who, interestingly enough, was a former head of science policy at Monsanto. And he had put forward the idea that in order to get genetically engineered foods out there, let's have a guideline, and he selected this guideline called substantial equivalence. Substantial equivalence was a guideline from 1976 that President Gerald Ford had put forward to accelerate innovation of medical devices. So to just give you some background here, if I were an inventor and I created a really great stethoscope or a new type of measurement device to be used in the hospitals, I had to get that approved by the FDA. That process would typically take around 7, 8, 9 years to go through an approval process. And let's say after I got it approved, I changed one little thing. Maybe I changed the knob color, or changed the color of it in some way, or some other minor modification, which really didn't affect the overall function or the safety. The issue was that person literally had to go through that again, 7-year, 9-year process. So what Gerald Ford said, to accelerate American innovation, that there would be a policy called substantial equivalence. As the word means, if the medical product A, which you did a itsy-weeny tweak to, was same as the new medical product, you did not have to go through the long 7-year process. You could fast track it. And in this process of substantial equivalence, it was part of, this is the actual document at the time, the 510K program for evaluating substantial equivalence. The policy was that you, as the reporter or the creator of this technology, do self-reporting, and there's a safety consultation done by the FDA based on what you give them. So when it came to GMOs, Michael Taylor said we should use this same policy. Now, just to be clear, by substantial equivalence, let me just go over here, the manufacturer should clearly identify the technology characteristics for each device, in this case a medical device, individually. So you get to choose what are the characteristics of the device that, or the system that you want to compare to your itsy-weeny change. And you decide if they're equivalent or not, plus or minus 20%. So if you had a stethoscope which was black and the new stethoscope was white, obviously the stethoscope has various parts, but if you just said, I want to use the color characteristic, you would say, hey, only this has changed relative to many other kinds of things, and you could get that allowed. So bottom line is, this was self-reporting. That means the manufacturer selected the criteria. So when it came to GMOs, Michael Taylor said, why don't we allow the manufacturer of genetically engineered organisms to use substantial equivalence? Now, forget the fact that a organism like a tomato is vastly, probably 1 billion times more complex than a stethoscope or a device. Think about all the interconnections, all the genes or the proteins. But the idea was, if you could simply inform the FDA. By the way, the FDA took a hands-off approach, but you could, for example, take the nutritional value, the protein, the fats or oils. You simply tell the FDA that there's equivalence. So if you and I, for example, created for example, a new company to create a genetically engineered blueberry, we simply tell the FDA in our own labs, we did the test comparing the color of the genetically engineering or color, let's say the size, the water content, 3 characteristics with the non-genetically engineered one, the organic one, and they're about the same. And when that goes to the FDA essentially issues a safety consultation letter saying, thank you, Bill and Bob, for letting us know that you've done this work. They don't even want to see the data. There's no even requirement. So I just want to make this clear. Based on that policy of substantial equivalence, the product could go to market. It's very, very loosey-goosey, all based on self-reporting. So in fact, there's no standards for doing lab research on the GM versus non-GM. There's no standards for distinguishing the material difference between GM and non-GM at the regulatory level. It's all very hands-off by the FDA following this equivalence model of substantial equivalence. So when all of this was going on, I, as a systems biologist with my PhD training with the development of CytoSolve, said, could we actually identify characteristics that were the characteristics one should use? So for example, we took soy, and what was going on at this time was we got in touch with a esteemed professor of plant sciences by the name of Don Huber out of Purdue. And the hypothesis, remember in science you see this observation, people are reporting there was a difference in this case with the corn. We wanted to find out what was that material difference. Huber gave us an insight, which was that the genetically engineered soy was engineered from the bacterium to resist the herbicide glyphosate, and by augmenting the soy plant with another immune system. So let me repeat again. So remember, the soy plant, by the way, 97% of the soy in the United States is genetically engineered. I'll repeat that again, 97%, only 3% organic. So Monsanto owns Roundup Ready soy, which means a soy that can withstand their own pesticide. And the way that they did this was they took a bacterium and they inserted it into the organic soy plant. And the hypothesis was that new soy plant was modified so it could withstand their own herbicide. And we were given an insight by Huber that this was done, which could affect the system of the C1 metabolism pathway. What is C1 metabolism? So what we did was we said, let's take the soy plant. And by the way, the C1 metabolism pathway, which is not only in soy, but it's in every plant, fungi, and bacteria, it's a biological system. It's a series of chemical reactions. So C1, capital C, number 1, metabolism, is a large-scale molecular reaction. It's almost like an engineering system that shows up in soy plants. It shows up in not only plants, bacteria, and fungi, the bacteria even in your gut. And so we said, could we actually use CytoSolve to explore the literature to actually understand all the molecular mechanisms, which means all the chemical reactions involved in C1 metabolism. So the C1 pathway, just to give you an idea, it involves a transfer of carbon units, and it's vital to the metabolism of a cell. So C1 pathway in plants includes the removal or detoxification of formaldehyde, which is a toxic intermediate in photosynthesis. So when photosynthesis takes place, formaldehyde is actually produced in the plants, but through a detoxification process within the C1 metabolic pathway, using a very important antioxidant called glutathione, using glutathione, the formaldehyde is removed. So when photosynthesis takes place, C1 metabolism is involved, and at a high level, you can think about formaldehyde is produced as an intermediary toxin, and the plant's nature has an amazing way of removing that toxin using glutathione through a process. So we said, could we really explore this pathway? First of all, understand what is involved, how this gets broken down and the process. So we literally went through the literature, using Google and PubMed and ProQuest and ScienceDirect. These are all publicly accessible tools. And we went through a whole process to find various articles that were involved in there. We looked across all different plants, and corn had a lot written in it, soy, but we knew that the commonality was a C1 metabolism pathway. Relative to this, we found over 11,000 scientific papers, and in those scientific papers, they related to close to 6,529 different experiments across 184 scientific institutions across 23 countries. So the point is that we just didn't cherry pick one paper. With CytoSolve, this very powerful technology, we were able to, and I'm showing you sort of a funnel diagram here, went across over 11,597 papers to identify 6,529 experiments in 184 laboratories in 23 countries. No one had ever done something extensive. No one could say that we were cherry picking. From those experiments, we identified various molecular reactions, like solving a big jigsaw puzzle. Each of those papers have little pieces of the C1 metabolism pathway in them. And then from that, we extracted those pathways, and then we started mathematically modeling each one of them using CytoSolve. That resulted in data, which we then used to publish a series of papers, which I'm going to walk you through. And the net of what we showed was that the genetic engineering of soy actually disrupts major molecular mechanisms across this system. And if the FDA had required real testing, if the FDA had required that they have real criteria, and they use the criteria of glutathione, which is a very important molecule, as I said, that we discovered that glutathione levels are about 250% less in the genetically engineered version of soy versus the organic soy. And in fact, this we showed through the mechanistic modeling of CytoSolve, and then we also validated, because we were fortunate to find work that was done actually where people grew the soy plant in a greenhouse, and our results matched. So that's following the scientific method, but I'll get more in detail. So we did a series. So the first 4 series of papers, which I'm going to walk you through, was really done to, we did it one by one because we didn't want to get attacked that we were just doing haphazard research. So we did it in a very methodical way. As I mentioned, we went through this 11,597 papers written on C1 metabolism. Out of that, we identified from those 6,597 experiments across those papers a relevant set of around 216 papers, which gave us 62 studies, and we discovered 3 important major mechanisms involved in C1 metabolism. I don't want to bore you, but at a high level, again, you can listen to this on podcast or video. On the video, I have some very nice diagrams if you want to follow through, but I'll also describe it. But think about an engine, and this engine is a C1 metabolism pathway. The engine starts at any point where you have something called methionine biosynthesis, where methionine is synthesized. That leads to what's called a methylation cycle. And in this process, formaldehyde is produced, and there's a third cycle called formaldehyde detoxification, where the formaldehyde is detoxed. So you have this very nice 3-part process: methionine biosynthesis, methylation, and formaldehyde. Again, these 3 parts form the C1 metabolism pathway. And just to let you know, this occurs in all plants, all fungi, like in mushrooms, molds, but it also occurs in our own gut bacteria, in bacteria. So we did this, and we published this in a journal, in an open source journal. And these were, by the way, the GMO research, just like the climate change research, is so politicized that we decided to choose open source journals. The major journals have already taken a position they're safe. In fact, just to let you know, Randy Schekman, who won the Nobel Prize in Medicine, who used to publish in the major journals, and I've published in them, he talked about how the major journals, there's insider trading. And when it comes to controversial things, they don't like to publish you. So we went to peer review open source, which means we can publish it to the community versus it being held back and manipulated. But what you can see here is the formaldehyde detoxification pathway involves various reactions involving a very important thing called glutathione. So glutathione is extremely involved in detoxing formaldehyde. If you do a search on Google on glutathione, you will see that it's called the mother of all antioxidants. The methionine biosynthesis pathway involves the use of formate, which is a product of formaldehyde, and the methylation cycle. Again, we documented these in a very detailed way, and this was published in the first journal. The second journal we did was, this was published in, after we published that in a Journal of Agricultural Science, the second paper we published was, now we had all the reactions mapped out. We said, let's use CytoSolve now to mathematically model it. And this was published in the journal, American Journal of Plant Sciences, and it's called in silico modeling. In silico means on the computer, in vitro by the means test tube, in vivo means in an actual body like of an animal where you do testing. But here we're doing testing in the computer because we've mapped out all the reactions. The reactions are invariant. And through this process, we took this 3-part process and we went through all the reactions. And because CytoSolve is this proven capability we had, developed since 2003 to 7 out of my PhD work, tested for another 5 years, we were able to use this very powerful engine, CytoSolve, to mathematically model the normal case of what plants do. And lo and behold, CytoSolve predicts what you see in the normal world, meaning that glutathione levels in a normal condition stay at standard levels, which means normal plants, non-genetically engineered plants. Glutathione levels maintain in a steady state, which means they don't go up or down appreciably, they don't get depleted or accumulate. And similarly, formaldehyde is produced and it's evanescently, which means temporarily occurs but then goes away. So these two things that we publish, the mathematical outputs from CytoSolve matched what people were seeing in vitro in other studies, which means glutathione is maintained and formaldehyde is created and it's taken away. So that was our second paper that we published in the American Journal of Plant Sciences, and we did sensitivity analysis to show under various conditions we were still getting the same process of formaldehyde being detoxified. When we looked at this C1 metabolism pathway, we also recognize that plants also undergo what is known as stressors. So remember, the first research we did was to map out all the pathways that was published in the Journal of Agricultural Sciences. Then we used that data to then do the modeling, and that was published in the American Journal of Plant Sciences, where we nicely showed that our model was able to predict what nature actually shows. So concurrence between the model and the actual observation. So that's what makes a model real. By the way, we're not seeing that with climate change. The models are not showing what's going on in nature. Here, the measurements are actually being validated. So the next step we did was we said, okay, the C1 metabolism pathway is also involved when plants undergo stress. So for example, that's called oxidative stress. So we said there was other research showing that when a plant undergoes stress, that it may actually lower its glutathione level because it's under stress and it's under oxidative stress, more formaldehyde may be produced, so it needs more glutathione antioxidant to detoxify itself. So you can think about that if you're under a lot of stress, research shows you produce more reactive oxygen species because you're undergoing stress, and that's why it's probably good to take of your health and take more antioxidants. So similarly, we went through the same process, went back to the literature, wanted to understand the mechanisms of oxidative stress as we had done it for C1 metabolism. So here we discovered various pathways involved in oxidative stress. Lipid peroxidation, which leads to reactive oxygen species, which then interacts with ascorbate glutathione pathway. Essentially, these three other pathways that are involved in the process of oxidative stress. Again, this is in plants. As you can see, glutathione is needed to address the reactive oxygen species, ROS it's called, when you have oxidative stress. So again, we modeled that and we interconnected. So we independently did the research in oxidative stress, validated that, and we said, okay, now you take the normal case of C1 metabolism, and what happens when you interconnect these two systems? Again, CytoSolve allows us to do this because we can literally do like Lego block building. And again, we went back to the mathematics, understood all the chemical reactions, and lo and behold, we again see our mathematical model predict what's going on in nature. We see as the plant is undergoing oxidative stress, by the way, which could occur in a drought or pollution, what you notice happens is that the plant depletes its level of glutathione. So that means glutathione goes down from its steady state level to a new level. And similarly, what happens as a result of that is, as you would think, formaldehyde accumulates because now you don't have enough glutathione because a plant is dealing with stress. There's not enough formaldehyde to do the detoxification process. So let me just be clear on that. Is that when a plant's under stress, or when you're under stress, this is why it's important to take care of your bodies or when you're under stress, is that your glutathione antioxidant levels drop to take care of the stress, and you might be taking away the glutathione from other processes. And the plant, that's what's actually happening. The C1 metabolism process is depleted of glutathione for formaldehyde accumulates. Now, the good news is that for plants, droughts obviously don't take forever, and if they did, the plant would die. But if they occur temporarily, and the plant's able to come back, so you have a temporary dysfunction of formaldehyde or lower glutathione depletion. So that's what that research showed, and we, again, tested it across different conditions. We did sensitivity analysis. As you can see, we consistently see glutathione level going up, but we see the accumulation of formaldehyde. And that was published in, that was our fourth paper we published, which says, do GMOs accumulate formaldehyde and disrupt molecular systems equilibria? Systems biology may provide answers. And in this paper, we asked the question, does genetic engineering do perhaps what oxidative stress does? That was a question that we asked. So we knew, again, from paper 1, that molecular mechanisms of C1 metabolism. From paper 2, we had understood how genetic engineering or how oxidative stress, which means when a plant's, paper 2 was where we did the mathematical modeling. Paper 3 is we understood with modeling what happens when oxidative stress takes place to a plant. And we showed there that the normal levels of glutathione level go down and formaldehyde accumulates. In paper 4, we said, now could we use the results of paper 1, 2, and 3? We were essentially building a very nice foundation here to understand this phenomenon. Could we actually understand what happens when we did the genetic engineering? And this led us to really understand what is the difference between soy and non-GMO soy. And as we went back to the literature, there's a lot of other scientists who've already done great work. We found another 107 papers. Out of that, we found 34 relevant papers with 11 different studies, which showed us 4 major differences between genetically engineered soy and non-genetically engineered soy. In fact, these researchers showed that 4 enzymes were at higher levels: catalase, superoxide dismutase, glutathione reductase, and ascorbate peroxidase, and 1 reactive oxygen species, hydrogen peroxide. So basically they said that the genetic engineering was perturbing those 5 chemicals. Now remember, those 5 chemicals I just said also show up in the C1 metabolism pathways. So that we had published in paper 1 and 2. So what we had was the puzzle was starting to come together, that genetic engineering was actually not doing something, it wasn't harmless, that it was actually affecting chemical levels in the plant. So let me repeat that again. What these papers showed, again, these are done by other researchers independent of us, that when you did the genetic engineering process, that it raised and affected, perturbed, disturbed the chemical levels of 4 other chemicals which are involved in C1 metabolism. So we had the aha moment where we said, look, why don't we take what their findings were, plug it in to the models that we had, and that's essentially what we did. So what we did was we said, okay, genetic modification, we had the oxidative stress pathways, and we notice here, if you look, is that there are, in the glutathione pathway, APX and GR, APX is, by the way, ascorbate peroxidase, and GR is glutathione reductase, were being affected. So in the glutathione pathway of plants from genetic engineering, two important chemicals were being disturbed. And we also noticed that the catalase, the SOD, and the H2O2, SOD's, just to show you, it's the superoxide dismutase, the APX, which is ascorbate, the catalase, and the hydrogen peroxide. Three other chemicals were being affected in the ROS pathway, the reactive oxygen species pathway. So I think from a layman's standpoint, that genetic engineering wasn't not doing anything. It was affecting the levels of other species, 5 chemical species. So we took those levels that were reported by other researchers in wet lab experiments, and we plugged it into CytoSolve, and we plugged that all together in one big computation, because remember, we built this up very slowly. Paper 1, paper 2, paper 3, and paper 4 was this paper we showed was that genetic engineering was actually doing something very similar to oxidative stress. Formaldehyde levels were going up, they were accumulating, and we also noticed, unlike the non-GMO case where formaldehyde was being created and cleaned up, in the GMO soy, formaldehyde levels were accumulating and glutathione levels were depleting, very much like what we saw in the oxidative stress case. So very different than the non-GMO organic case, glutathione levels maintained, but here glutathione levels were being depleted. And in fact, we noticed that they were going to a different level. It's almost like the plant was under constant stress. So to summarize, these 4 papers showed that genetic engineering does disturb the original state of the plant and puts it into a new mode of depleted glutathione levels. What we found was around 250% difference in glutathione, and formaldehyde accumulates. So our view was that if substantial equivalence had been applied to those 2 criteria, they would have been significantly different and maybe would have raised red flags. But the bottom line is the FDA doesn't really require any reporting. It's all self-reporting. And what we had discovered here was if we apply systems biology, if we apply CytoSolve, you can actually discover what should be the chemical criteria. Right now, the model is Monsanto chooses whatever criteria they want, which may have no relevance to really distinguish the difference between a GMO and a non-GMO. Now, when we published this work, we published 4 papers. We'd basically shown that genetic engineering, the genetically engineered soy was vastly different if you use the criteria of glutathione and formaldehyde than the non-genetically engineered soy. And this created almost like a bomb went off in the pro-GMO community. And we said in our paper, look, we're not pro or anti-GMO. We just wanted to find out what was going on. A researcher at the chairman of the Horticultural Institute at University of Florida, public university, which our tax dollars support, started attacking me personally, said I didn't invent email, calling me all sorts of names. And more importantly, he never attacked any of our really detailed research, didn't attack the chemistry, didn't attack the equations, none of that. And in his attack, he kept saying, I'm an independent researcher, I'm not paid by Monsanto. About a year later, and he said, these guys need to prove their mathematical models, not knowing that our mathematical models are actually based on actual experiments. We're not like making up any of these equations. And interestingly enough, a group out in out in California called US Right to Know did a FOIA on this researcher, Kevin Folta, because University of Florida is a public university. 4,000 emails came out, and I was in India at the time doing a debate on GMO and non-GMO between agricultural students. I was facilitating it. And at a few days after I finished that debate, on the front page of the New York Times, it came out that Kevin Folta, in his emails, it was revealed that he was actually a spokesman for Monsanto. He'd received a check for around $25,000. So Folta was exposed. But we actually wanted to do further research to further validate our work, because that's what scientists do. You're never satisfied. And that resulted, I'm going to go back to the science here, that resulted in a very fortunate case. And by the way, this was covered by major media, our research, it went viral all over the world. A lot of the mainstream US media didn't cover it because they were essentially supporting the old thesis that natural plant breeding is the same as genetic engineering. A lot of these people need to learn science. We in fact got statements from scientists, hundreds of scientists and MDs supported our research. They signed off on a petition that the FDA really should relook at this entire process and guideline. We got very fortunate, which led to our final and fifth paper, that there was research done by a group in Leeds in the United Kingdom where they had actually grown the soy plant in a greenhouse, and the organic and the non-organic. Remember, our research had predicted from all of their integration of the mathematics that all the integration of the mathematics had predicted that the glutathione levels in a soy plant would be 250% less if it was genetically engineered Monsanto, and 250% more if it was an organic. And these researchers, which I'm going to share with you, actually had grown the soy plants in a greenhouse, and they had come across something fascinating. They also found very similar levels. This was published in Plant Physiology, and we then took that research and we did the comparison. And what we found was they had, and this was published in, again, in the American Journal, I believe, of Plant Sciences. This was our paper which said in silico analysis and in vivo results concur, which means our mathematical models and the wet lab, in vivo means in the plant, concur on glutathione depletion in glyphosate resistant GMO soy. That's Monsanto soy or Roundup Ready soy, advancing a systems biology framework for safety assessment standards. And I'll just show you what the results we showed. We showed, they had found out the glutathione ratio levels in organic was 9.9, and in the transgenic Roundup Ready RR was 3.7. So that's the actual lab experiments. And look at what we predicted, very close. We predicted 9.75 or 9.749 and 3.9. So this basically showed that we understood the mechanisms why this was taking place. So beyond the wet lab experiment, we actually know why this is taking place. And the why is important because that's science. When genetic engineering of soy takes place, the C1 metabolism pathway is disturbed. Various molecules get disturbed. That disturbance leads to an oxidative stress type state, and the plant is essentially under constant oxidative stress. And based on that oxidative stress, it has lower levels of glutathione, higher levels of formaldehyde. And I would argue that the GMO, genetically engineered soy, is actually a weaker plant. And this will be a discussion of a future podcast in the interest of time. But many of the genetically engineered seeds are now coated with neonicotinoids. Neonicotinoids protect that seed from being attacked by soil organisms. Well, why do you need neonicotinoids? By the way, neonicotinoids are a nerve agent which destroy the bees. So what we're saying is, you took an organic soy plant, then you hit it with weed killer, glyphosate, and then you produce a genetically engineered version of the original soy plant because your weed killer was killing that. And then that new plant has lower levels of glutathione, and it's a weaker plant, and now you coat it with neonicotinoids, which are essentially affecting the bees. So this entire process, what I can tell you from a system standpoint, it's not politics, it's science. The science clearly shows that we don't have, at minimum, worst case, best case, we don't have safety assessment standards for genetically engineered foods. So are GMOs safe or not? The issues we don't know, and as far as we don't know, we should, in my view, there should be a moratorium on genetically engineered foods. And the data also shows that the poor black people in Africa and the poor brown people in India, it's not genetically engineered foods. They need more irrigation, they need more tools, the farmers need more education. That's what's really needed. And in fact, the destruction of the soil has taken place because of pesticides. So anyway, I hope this was valuable. I don't know if there's any questions coming in. We scroll down for comments. People said, Folta, outstanding presentation, thank you.
Oh, no bad for the bees, colony collapse disorder. Yeah. So I think the point I want to make here is the climate change. Look, you may consider me a lefty or a left winger for going against Monsanto, but I'm a scientist. But when I also expose the failure of the Paris Accords and the fact that CO2, demonizing CO2 as a thing that's causing climate change, which is a whole nother discussion, which we've talked about, which we can talk about, doesn't also correlate. The data, the models don't work. Our models work. We don't understand why, and we validate it. The climate change models do not work. And in fact, I think the climate change scare is actually taking away from the bigger, real green issues of what's going on with the bees, what's going on with pollution in the ocean, clean air, clean food. And we're forgetting about that. These are certainties that our research shows, other research shows. So if we want to really be green, let's go back into organic foods. Let's go back into locally grown foods. Let's put a moratorium on GMOs because there are no safety assessment standards. Anyway, this is Dr. Shiva Ayyadurai. I hope you've enjoyed this.